Hello everyone, it's me Mohammad Sulieman, and today I will talk about the cell cycle. Right, this article we will go throughout step by step how cell cycle is happening, and we will know about all the processes and the metabolism that happens during the cell cycle. So the first question that comes to our mind that we want to answer is, what is the cell cycle? When we say cell cycle, we mean that is a series of events that cell go through while it's growing and dividing. Every cell needs to grow and divide until or some group of cells they only grow and die, just like the blood cells, because blood cells they don't divide, they only grow up and do their function, and after a specific period of time then they go and die. All right, but in the cell cycle, we are talking about generally like tissue cells and the other cells, they are growing and dividing and so on. Even the blood cells inside the bone marrow, all right, some of them, they are dividing. For example, stem cells. Stem cell is dividing into two cells until it give us rise to another different type of blood cells. All right, so actually, there are 5 phases of a cell cycle. The first phase is called the primary phase, all right, or it's the primary growth phase. The second one is called the S phase or synthesis phase, where DNA replication is happening. The third phase is called secondary growth phase, all right, and it's called also G2 or secondary growth phase. Those 3 phases, they are considered as one stage and is called interphase. All right, so interphase includes G1, S phase, and G2 or the secondary growth phase. After that, when secondary growth phase is finished, the first phase is the mitosis phase, and mitosis is the process of the division of one cell into or single cell into two daughter cells. All right, so it's a start. Okay, when we are talking about internal separation, one nucleus is separating into two nucleus, and we will talk about all that later on. So, and the next one is the cytokinesis, and the last stage of the cell cycle is called, or the last phase is cytokinesis. And cytokinesis is when there is a total and complete separation of the nucleus and the cytoplasm. So here, if you look at this diagram, it shows us all the phases starting with mitosis, with G1 or primary growth phase, and then it goes for S phase, synthesis of DNA, then the secondary growth phase, and after that mitosis, and last thing is cytokinesis. And the cycle is repeating again and again and again until when? Until the body is dead or not functioning. That's all. All right, so during this cell cycle, what exactly happens? First, cellular growth. Cell is growing up, all right, because after the first or the last division, when there are two new cells, all right, then this new cell or daughter cell is starting to grow up, all right, and after that, then the genetic material is duplicated on the synthesis phase, and after that mitosis happens. All right, so when the genetic material is duplicated, that means what? The nucleus should be separated. Every cell should have its own nucleus because there will be two daughter cells. And after that, number 4, in cytokinesis, when cell is separated into two cells. All right, the first phase is a G1 phase or primary growth phase. Let's start talking about that. All right, it's the first stage on the interphase, and it's called the G1 stage or G1 phase. The first or the primary growth phase is the first growth stage after cell division. And in this stage, exactly, cell mature by making more cytoplasm and organelles. So the cytoplasm and organelles of this cell is grow up. So and after that, cell carries on its normal metabolic activities until changes occur. So and this phase or this phase, this stage what happens exactly? What happens is that cell is growing up in the cytoplasm, and also the organelles, internal organelles of the cell, like mitochondria and the other organelles of eukaryotic cells, as we know that, and until the cell became mature enough to divide. All right, so at this point exactly some changes starts to happen and to occur at the cell, which makes it go to the second phase. All right, those metabolic changes, they prepare the cell for the division, and at a certain point the cell is committed to division and moves into the S phase. All right, so the S phase, or the synthesis phase as it's called, this is the second phase on the mitosis, on the cell cycle, and we are talking about the interphase, the second phase on the cell cycle, and also on the interphase. And it's called synthesis phase because DNA is synthesized. At this phase, DNA is synthesized and replicated into two copies. So primarily, we have one copy of the DNA, but after that, we will have two copies of the DNA because of the synthesis phase. So we can look at this diagram. So while we have one DNA, and after that the DNA is being copied into two similar copies of DNA. So it have the same characteristics, right? But why DNA replicated, or why DNA is copied into two copies? Because this cell will be divided into two cells. So that means what? That means this DNA must be copied in order to for every cell to have one copy of the DNA. This is the original DNA, and this is the identical DNA. All right, the third phase on the interphase is called the G2 phase, or the secondary growth phase. In this phase, all the cellular components needed for cell division are produced. All right, just like what, like spindle fibers, all right, like centromeres and centrosomes. You know that all the organelles that are needed for the separation of the genetic material and the chromosomes into two nucleus will be produced here. And also above that, DNA will be checked, the DNA replication. All right, sometimes error might happen. When error happens, so it needs to be checked again to see if there's any error in the copy of the DNA. And the secondary growth phase, it occurs after DNA has been copied, as we said before, and all cell structures needed for division are made, just like centrioles and spindle fibers and so on. And after that, both organelles and proteins are synthesized. So at this stage, now the cell is ready for what? Ready for separation of the nucleus. All right, so the interphase, the first stage on the cell cycle, which includes the primary growth phase, the synthesis phase, and secondary growth phase, is all about what? About preparation of the cell for the separation or for the division. After that, the cell directly enters the mitosis phase, and we know that there are two types of a cellular division, all right, according to the cells. Either it's mitosis or meiosis. When we're talking about mitosis, we are talking about a division of normal cells, all right, just like a tissue cells, blood cells, or like a stem cell when it's dividing into two daughter cells and so on. But the meiosis, we are talking about the sperm, all right, we are talking about the fertilized egg. When a fertilization happens, all right, when a sperm is fertilizing an egg, and then after that a new infant is being produced inside the mother. So this is meiosis, while mitosis is just only the cellular division. Right. When we come for the definition of mitosis, it's a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cell. So chromosomes have already been replicated during what? During the S phase. And in this phase, the chromosomes, they must be divided, all right, and then every nucleus have its own DNA or its own genetic material. All right, so the replicated chromosomes are attached to mitotic apparatus that align them and then separates the sister chromatids to produce an even partitioning of the genetic material. So during this mitosis phase, what happens exactly is that the genetic material or the chromosomes, because we say DNA have been replicated. So the replication of this DNA, that means every chromosome will be copied and duplicated. So when mitosis is happening, all these genetic materials or all those chromosomes will be divided equally into two groups. Every group have the same characteristics, same information, and so on. And this separation of the genetic material is called karyokinesis. All right, and because it's just like when we say karyo, we always talking about the chromosomes, right? So, and kinesis means separation. So, when there is a separation or there is a division of a chromosomes, it's called karyokinesis. After that, this will be followed by the separation of the cell cytoplasm in a cellular division or the cytokinesis. But here in this phase, or the mitosis phase, mainly the division will be happening in the nucleus. The nucleus will be divided into two. All right. So even this mitosis phase have 5 stages. All right. It starts with the prophase. After that, it's happening prometaphase, and then metaphase, anaphase, and the last one is telophase. Before moving on to cytokinesis. So let's talk briefly about every one of those phases one by one. All right. The first phase is the prophase. And the prophase is when the nuclear membrane breaks down to form a number of small vesicles and the nucleolus disintegrates. All right, so I will repeat again so everyone understands what I'm talking about. First, I talked about the interphase, which includes the S phase. All right, the G1 phase or primary growth phase, and S phase, and the G2 phase or the secondary growth phase. Right. And we say during the interphase, what happens? Cell is being prepared for the separation of the nucleus. All right. And when we enter the mitosis, then in mitosis, during this mitosis, it goes throughout 5 stages. All right, the separation of the genetic material and the nucleus go throughout 5 stages. The first stage, which is the prophase, where exactly the nuclear membrane starts to break down, because the nucleus must be separated into 2 different nucleus. So this nucleus will be should be weak, or the nuclear membrane should be weak in order to be dividing into 2 nucleus. So the nuclear membrane breaks down to form a number of small vesicles and nucleolus disintegrate. Right? So we can look at the picture and the next slide, and then we talk about that. A structure known as the centrosome duplicates itself to form 2 daughter centrosomes that migrate to opposite ends of the cell. All right, and also in this stage, centrosomes will be duplicated. Actually, it's 1 centrosome, but in this prophase, the centrosome will be duplicated, and each one of the centrosomes will move to and opposite side of the nucleus. So now the nuclear membrane is weak, and also the centrosomes have been duplicated and moved to the sides of the nucleus. And also during this phase, the centrosomes organize the production of microtubules that form spindle fibers. Also, the spindle fibers are being produced here. And what is the function of spindle fibers? Spindle fibers are needed for the catching and separating the chromosome. All right, because when there's a full chromosome, it's called chromosome, and single arm of chromosome is called chromatid. All right, so we want to separate every chromatid into a single nucleus. So the chromosomes condense into a compact structure. Each replicated chromosome can now be seen to consist of two identical chromatids, or a sister chromatids as it's called, and then they held together by a structure known as a centromere. If you look here, we have the centrosome, and every centrosome it have centrioles. This is the centrosome and the other centrosome, and those are the spindle fibers. All right, so during this phase, this is what happens: production of those centrosomes and production of spindle fibers. And as we can see here, this is the full chromosome, and one arm of chromosome is called chromatid. And also the nuclear membrane have been fragmented, or it's been weakened, so it will be easy for the nucleus to divide. Generally, this is the nucleus, and those are the characteristics of the first one, which is prophase. After the prophase, we move directly to a pro-metaphase, and on the pro-metaphase, the chromosomes, led by their centromeres, migrate to the equatorial plane or equatorial line in the midline of the cell, at right angles of the axis formed by the centrosomes. So all the chromosomes will line up on the center of the nucleus. For what? For them to be separated into two chromatids. So when the chromosomes are lined up, after that the spindle fibers bind to a structure associated with the centromere of each chromosome, and it's called kinetochore. And this kinetochore is the area where spindle fibers are being attached. If we look here, all right, so this is the chromosome. Those are the chromosomes, and one arm we said is chromatid, and here it is a kinetochore where the spindle fibers are connecting to the chromosome. So every spindle fiber will connect to one arm of the chromosome, and after that, preparing it for what? For separation, because every spindle fiber will pull up, and then the chromosome will be separated. All right, so the individual spindle fibers bind to a kinetochore structure on each side of a centromere, and also the chromosomes continue to condense. And if you look here on the pro-metaphase, this is actually a photo explaining that the chromosomes are lining up, and those are with the green lines are the spindle fibers that are connecting to the kinetochore of the chromosomes. And here we can see clearly, or very clearly, that chromosomes are being in the center and spindle fibers are connected to the kinetochore on the chromosomes. After that, when spindle fibers have been connected to the chromosomes, the cell moves to the next stage, which is called metaphase. And the metaphase, in this stage, the chromosomes align themselves along the metaphase of a spindle apparatus, and guess what? The pull up of this spindle fibers will start. All right, as we can see, all of them, they are lined up, and the spindle fibers are now pulling up. And when a pull up is happening, the last phase is coming, or the first phase is the anaphase, where it's considered as the shortest stage of mitosis, and the centrosomes or the chromosomes are divided, and the sister chromatids of each chromosome are pulled apart, or they disjoint. All right. And then we can see here all of them, they have been disjoint. Now we have a separated chromosomes. Each set of the chromatids have been moved to one side of the nucleus. And by that comes the telophase, and telophase is the last stage on the mitosis, which is the nuclear membrane reform around the chromosomes. All right, so now after the chromosome or the chromosomes have been separated and pulled apart to on each side of the nucleus, the nuclear membrane itself must be separated. All right, in order for what? In order to have two copies of a nucleus, but they are identical, they have the same genetic information, and they have the same characteristics. As we can see here, now this is a nucleus, and also this is a nucleus. Every nucleus have the same amount of the genetic material or the chromosomes, and also the cytoplasm of the cell starts to be like it's wanted to separate or starts the separation, but until now it's not separated, because the separation is happening on the last stage of the cell cycle, which is the cytokinesis. And on the cytokinesis, this is where a complete separation of a cell is happening. If we look, the final cellular division to form two new cells. So here, after a separation of the chromosomes and separation of the nuclear membrane, now comes the cytokinesis, where the separation of a cell, because if we come for the name cytokinesis, cyto means cell and kinesis means what? Separation. So here the cytoplasm is separation. And as we can see here, cytoplasm is completely separated, and we have two different cells, daughter cells, with an equal and the same characteristics of the mother cell. And here it's just showing us all what happens during the cytokinesis, where the cytoplasm is starting to tighten up and up until it's separated into two cells. So far, this is all what happens for the cell step by step, starting from a single cell and ending with the two daughter cells. And thanks for watching. It was a pleasure for me, and I hope that everyone got my explanation well. If you are interested more in my classes, and you can follow me on my Facebook page, and also I have a YouTube, and you can subscribe. Don't forget to subscribe and share with your friends, and see you in the next lesson. Bye bye.